restriction enzymes, or restriction endonucleases, or molecular scissors, found in bacteria, provide defense mechanism against invading viruses by degrading viral DNA without affecting bacterial DNA. And each bacterium has its own unique restriction enzyme, and each enzyme recognize only one type of sequence, so DNA sequence, that recognized by restriction enzymes, called palindromes, which are sequences, read same on two strands in opposite directions, as mentioned in next slide. The mechanisms of restriction enzymes start by scan DNA, then bind to DNA to recognize specific sequence. After that, cut each sugar-phosphate backbone of DNA by hydrolyze phosphodiester bonds, as mentioned in next slide. Continuously. This slide show cleavage mechanisms of restriction enzymes, as we show the first example, refer to sticky ends 5 overhang, while the second example refer to sticky ends 3 overhang, whereas the third example refer to blunt ends without overhang. So, sticky ends restriction enzymes, called also cohesive ends, because they are useful for DNA manipulation, and can be converted to blunt ends with nuclease or polymerase, while blunt ends restriction enzymes, useful for certain types of DNA cloning experiments, and can be converted to sticky ends by ligating to synthetic adaptors. This slide referred to the common groups of restriction enzymes, as we show. The first group refer to sticky 5 ends overhang, while the second group refer to sticky 3 ends overhang, whereas the third group refer to blunt ends without overhang. Restriction enzymes have 4 types, as we show. Type 1. Cleave DNA at random length from recognition sites, while Type 2. Cleave DNA inclusive recognition site. So, Type 2. Useful for gene analysis and cloning. On the other hand, Type 3. Cleave outside recognition sequence. So, it require 2 recognition sequence in opposite directions within same DNA. Whereas, Type 4. Cleave only modified DNA in approximate 30 base pair away from sites. For restriction enzymes mapping, must digest DNA with restriction enzymes, then resolve digested fragments by gel electrophoresis, and the resulted bands on gel refer to number of restriction sites, while the size of bands refer to distance between restriction sites, as mentioned in next slide. Continuously. This slide show example of genomic DNA digest and plasmid DNA digest by restriction enzymes on gel electrophoresis. Finally, restriction enzymes used in gene characterization by RFLP markers, and gene mapping such as restriction maps, and gene manipulation such as gene cloning, and protein expression experiments.